Thank you very much, and thank you for testing my wits and seeing if I can speak at 5 a.m. I was asked to make a talk that will be general and accessible to people who don't know biology, and then to people who don't know computers, so I think what you'll get is a mishmash that caters to no one. Life is information processing itself. We could view ourselves as computers. Our DNA, a code of 6 billion letters, is transmitted from generation to generation, passing on the information of life. This information is actually a code for circuitry that makes our body from a single cell into this walking, talking body, and having each cell be able to sense its environment, make decisions, and you could view cells as cell circuits. Now, what happens in disease is both the code itself, the DNA, and subsequently the circuitry gets messed up. And what some of big data is trying to do is trying to understand all of this. So zooming back from the macro, human beings and why they get sick, what system does it occur in, what's molecularly going on, how the genes and the proteins change, how does this connect back into the DNA, and big data, or big data from computational biology bioinformatics perspective, is going the other way around. Looking at the DNA, looking at other molecular measurements such as RNA, and trying to figure out how and what goes wrong, DNA to phenotype. In cancer, it's a little bit different. We don't have population forces and evolution of a species, but rather we have a single greedy cell evolving. So you have somatic mutations that are, where's this like taser-like pointer? You have somatic mutations that cause alterations, driving mutations that change the cellular processes. And the end goal of all this big data, as was mentioned earlier, is how could we use this understanding to actually impact patient care, to actually make human beings be treated better and more effectively. So big data actually did start in cancer a long while ago from very simple technology. This was gene expression, the ability to suddenly, in one experiment, measure something like 8,000 genes. And the first big news was this paper from 1991. It actually is what got me, as a computer scientist, to step in and become a biologist. And it basically showed that looking at genes, looking at a molecular signature of how genes are expressed, we could distinguish between AML and ALL, two clinically very different cancers that require very different care, yet pathologically couldn't be distinguished before we went to look at the molecular level. There, using supervised classification techniques, they could pick up a gene signature of 50 genes that was predictive of whether you had ALL or AML very accurately. Again, no single gene, but the signature of 50 genes. Unsupervised approach to actually discovered new subtypes of cancers. The first case in B-cell lymphoma, later in breast cancer. We're actually in an unsupervised clustering approach, finding more and more subtypes of cancer. These have different clinical outcomes, require different treatments. And on, using just microarrays and gene expression, there have been a lot of tools to identify subtypes of cancers, predict response, predict which therapy should be used, just using gene expression for microarrays. Now, big data came to biology the reason they say big data in biology is because of this picture right here. What you see is an old gene center based on Sanger sequencing of once upon a time, a long time ago. You see this entire floor? This little machine right here in 2008, in a single day, can sequence on this tiny desktop what the entire floor took to sequence in a month. That's the next-gen sequencing revolution, and that's its size. An entire floor full of machines, people, personnel, and air conditioning could now be replicated into a single tiny hand-operated machine in one day. But the revolution didn't stop there, so here are the specs. So these things read out sequences, read out DNA, basically read out this 6 billion letter code. And these are the specs in 2008, and they continue to grow. The number of reads that you can sequence in an experiment, the length of these reads, how many letters can you sequence in a single shot, the throughput, the cost, and this is pretty close to the current specs, maybe 8 months old. And it rapidly continues to evolve, we're able to generate more and more and more sequence data at a cost-effective manner. And again, this is a favorite plot. This is Moore's law, which was considered super exponential. And this is our sequencing costs, creating a tsunami of data, trillions and trillions and trillions of reads of RNA. Importantly, it's DNA. It's not only DNA. We have a lot of different protocols. So we could look at our DNA, we could look at our RNA, we could look to what binds to our DNA, what binds to our RNA, what bits of RNA are currently being translated into proteins, and the number of new protocols to actually use DNA to query different types of biological questions is rapidly increasing. So again, for those of you who don't know how this works, think of it, we have this long piece of DNA. We can't read it as is. We need to break it up into little bits, shear it up. We need to amplify it, make many copies of it, and now we can read these little bits and put it back together again. Basically, now that we have these short reads, currently about 100 base pairs, 100 letters long, we can begin to try and map them back to the genome and see what they say. That's DNA. RNA is a big application of this. Here, we don't only want to read the letters, but we want to quantify the abundance of each gene. So here up here, you see the RNA being expressed. Different genes here are expressed at different levels. Each squiggly here means this is expressed more than this. RNA-seq is actually taking little bits of it, sampling the transcript out there, sampling little bits and pieces, and then, as I showed you earlier, mapping them back, each of these things, the letter, mapping them back to their respective pieces, seeing that, we have many more transcripts here, less here. This is actually being spliced, so only this part and this part of the gene is being expressed, so we could actually look into transcript structure. Now, this is all very easy. All you need to do is map it back. Not quite. These reads are rife with artifacts, systematic biases. If you just look at them naively, take regular error models, nice looking statistical distributions, you're going to get huge artifacts. And in fact, even when you have a pipeline that works, some of these steps, some of these complex molecular steps can abysmally fail, so you need a lot of QC. You can't take this data face value. You can't analyze it naively. If you're getting into this game, you have to think very deeply, understanding the molecular protocol that generated these data and what step and what type of bias and artifact each of these protocols can create. And one of the big ones is an exponential amplification of the material really helps. So you really need to know what you're doing. And actually, the next speaker is the biggest expert in the world. He has some of the best algorithms out there. He's not going to speak about this, he told me. So if you want to get into this game and you're not into it already, read all his papers. And if you are into it and not using his methods, we compared it. They're the best. Okay, so what is cancer? Cancer is basically this code, this DNA, and it has mutations. It's evolving, and these mutations give it many tumorigenic abilities to rapidly grow out of control, to invade new areas and places it's not supposed to be, to evade the immune system, to live under different metabolic conditions and all these other tumorigenic abilities that it needs. And importantly, the DNA correction machinery has gone awry, so you have many, many mutations. So to try and understand this cancer and how alterations in the DNA actually give the cancer all these tumorigenic phenotypes, what are the alterations and how do you get a cancer cell? The ICGC, which was nicely introduced before me, was brought about. And it was a very optimistic time. We were going to sequence a lot of tumors. We were going to look at their DNA, their RNA, their epigenetics, their methylation, and we were going to understand everything. And for each cancer, prostate, esophageal, we were going to basically characterize the entire molecular landscape, understand everything we need to know, design all the necessary drugs, and cure cancer. And what we discovered is that this is actually a lot more complex than we thought. Each cancer has many more mutations than we thought, and the variability or the landscape between cancers is much more complex and dramatic than we had thought. So even asking the simple question, what are the mutations? What are the changes in the DNA that actually give the cancer these tumorigenic abilities? What are the mutations that matter? What should we target to try and shut down the cancer cell? And to distinguish that from all these mutations that just are there because of this quality that cancers mutate and the genome integrity and DNA correction machinery has gone awry, that's become a huge challenge. And computation is a bottleneck. It's like finding a needle in the haystack, and what's completely confusing is that the needle in the cancer genomics case actually also looks like a piece of hay. It's often very indistinguishable. And what people did at the beginning is simply use simple statistics. Does a mutation occur more frequently than would be expected by chance? And here I'm going to give a little plug for data integration and tell you a little bit of some things we did. The problem is that you just have so many different mutations. In sequence mutations, there's only 6 mutations that occur in more than 5% of the samples. Copy number alterations, you have more. These are big chunks of DNA, sometimes including dozens of genes that are amplified or deleted, but then you don't know what gene is being targeted. In about 70% of the regions in breast cancer, we don't know the target. And this is after we've collected thousands and thousands, about 3,000 breast cancer samples to try and understand this. And the trick is that I'm actually an imposter, and all us cancer biologists up here are going to be imposters when we try and call ourselves big data scientists. Big data is really about big data. It's really about getting billions of samples. If you're looking at facial recognition and image recognition or social networks or recommender systems for what should you buy next, they really are looking at billions of samples, billions of examples to learn from. And while we have billions of reads, each cancer has more than 100 billion reads. And if we count reads, we feel that we have a lot of big data. Certainly, Amazon Web Services are really draining my budget in terms of analyzing these billions of reads. At the end of the day, these billions of reads represent only typically 1,000 cases, 1,000 samples per each cancer. So actually, in our world, while most big data and machine learning have a very small number of variables and then this huge billions numbers of samples, so you really don't need to do much. In fact, in the big data with all the deep learning, now the game is who has the bigger computer, and the challenges become air conditioning rather than algorithms. Fortunately for me, as an academic who can't compete with Google on hardware, I actually have what to do. So my world, where we have many variables, tens and even hundreds of thousands of variables, and a very small number of samples, hundreds to at most thousands of samples. So we're really underdetermined. We really don't have enough data. We don't have enough examples to try and learn and understand this very, very complex biology. And one of the things that we have at our disposal is actually to try and insert domain knowledge, to insert some intuition into the biology, to do some data integration. And I want to give you 2 examples of this in the sort of driver versus passenger challenge that I posed before. Here's an example that we did in 2010. Again, previously people just counted how many mutations they have. And we made 2 assumptions. We said, well, if it is a driver mutation, if it's causing tumor genotype, we should see that the occurrence of a driver should co-occur with a phenotype that we can measure. And what type of phenotypes that we can measure? I mean, there's the tumor gene phenotypes that we'd like to measure, but we have gene expression. And because of these signatures I showed you at the very beginning, almost any cellular or phenomenological phenotype will express itself as a gene signature, as dozens of genes that go up or down. So we can actually try and associate the occurrence of the driver in the DNA with one of these gene signatures. And the other trick is if you amplify DNA, you don't change the gene at all. The driver event is actually trying to raise the expression of a gene. There's many ways to raise the expression, how frequently a gene is being expressed and what abundance. Only one of them is making more copies of its DNA. And if the cancer wants to overexpress a gene, it's a very powerful system with a lot of evolutionary innovation. It's going to find other methods. So we actually want to associate the expression of the gene with the phenotype. We have the DNA to get causality. We then associate expression to phenotype. And that's how we could really do the first big attempt to pinpoint drivers, to find these haystacks. In the next step, and this is really data integration and biological intuition, to build a computational score and search procedure. Another example of integration, you can view cancer here as a plane wreck. We have a lot of mutations. We don't know which are the early ones, which are the late ones, which caused this wreck. It's hard to find in this wreck what caused it. This is primary tumors. On the other hand, we have a lot of tools to perturb, to knock down each and every gene using shRNA or the newer CRISPR technologies. But this is typically done in very artificial situations without being in a human, without the immune system, without the microenvironment, in a dish, and in very small samples. And it's also very noisy technologies. And so we'd like to get the best of both worlds, a pinpointed targeted perturbation assay, plus the primary tumor, what really happens in humans, and integrate them both together. And this is work by Felix Sanchez-Garcia. He's down the road here at the Guardian doing data science for you today. Unfortunately, science is not a very lucrative option for young scientists, and he has a family, so he left science, but he's one of the biggest experts in cancer genomics, so that's unfortunate. Again, intuition, what features can we use? You don't want to just do feature selection blindly, look at, let's say, all these reads, all these genes. You have to think, what is the driving biology here? And here one of our strongest features from the cell lines is what we call oncogenic addiction. And that is we're looking for cases where when the gene is expressed at normal levels, the tumor doesn't care. But when the gene is overexpressed, when it's expressed well above normal levels, that can predict that that gene is a vulnerability for the cancer. And if the gene is overexpressed in expression, when you knock it down, the tumor will lose its viability. So we're looking for this signature. And this is one of the many features that we put into our algorithm, our classification algorithm. And here's a little bit of real data. So you can see this is a real oncogene. And the gene right next to it in the genome, as amplified, doesn't have this. Again, these two examples of how this signal really can pull out the drivers versus the passengers. And of course, we put all this together in a hierarchical-based classification algorithm with a lot of your standard favorite machine learning tools, particularly transfer learning, so we could take very strong drivers and use them to learn distinctive features of some weaker drivers. And using this approach, we got really incredible amount of accuracy and predicted 11 out of 12 novel drivers in cancer. Taking a step back, beyond cancer, basically DNA really does distinguish our phenotype for almost any disease, not only cancer. And what we've found in the general genetics field is that almost 90% of the loci that explain or link or associate with disease, they aren't in the genes, but they're outside of genes, and a lot of evidence now shows that they are in regulatory region. So that a lot of the variation between us, not only in cancer, but also what makes me, and you, and why we're different, including in our disease susceptibility, is actually changes in RNA, the regulatory network. And in order to link between DNA and phenotype, we really need this RNA as an intermediate. It's more complicated than that, and I want to share with you how complicated biology is. DNA is single code, but it actually encodes for many different cell types. And when disease goes wrong, it goes wrong in a particular cell type, often a small cell type. For instance, neurons in autism, adipocytes in obesity and in other metabolic syndromes, immune cells, particularly dendritic ones in Crohn's disease. So we really need to look at the tissue. And when we find a genetic association, pinpointing, okay, at the level of the DNA, we can see that this gene matters. Where does it matter? In what system? In what tissue is often very unclear. So what we have is one genome, 3 billion letters. And most of the data that's being sequenced out there is DNA. But this data now needs to express itself in many different cell types. Only 1% of these 3 billion base pairs are expressed as genes. These genes differ across the cell types. And people thought, okay, at least we could look at the regulatory regions, but now new epigenetic data, again, big data, has allowed us to learn that between tissues, even the regulatory region that tells how the gene should be expressed is changing between each of these cell types. And they could be very different for each gene. They could be far away because DNA not only has a linear structure, we don't only have a sequence, but it can fold. So you can have a regulatory region and a gene very far away that fold in 3D structure and influence one another. So what I'm trying to get to you is this is very, very complex, and gene expression is the key to understanding disease. And finally, I'd like to say what we should be doing going forward. We should actually look at gene expression at the single cell level. We need to characterize our cell types. There's about 300 characterized cell types. Immunologists will say there's 300 immune cell types. T-cell biologists will say there's 300 types of T cells. We actually don't know our cell types. We've made a map of our DNA, the genome map. Now we need a cell atlas. We need to characterize all our different cell types in the body in order to know where each and every mutation goes wrong, what cell type does it affect. And what makes this now possible is a new technology, and I'm telling you things that were published just 2 weeks ago in Cell. This is the in-drop technology where you can use tiny microfluidics. And this allows you in a cost-effective and time-effective manner in a single experiment to assay single-cell RNA-seq and look inside individual cells and get their expression profiles for about 1,000 cells in single experiments for around $4,000. And there were 2 papers published in Cell, slightly different from each other. Both of them were basically bar-coded reads. So you took that read I was talking about before, and you first put a cell barcode into it. You say, what cell does this read come from? So now when I have all these reads, I can actually pool them and say this read came from cell 1, and this read came from cell 9021. And another very important thing is a random molecular tag. What molecule did this come from? Because when you're amplifying in exponential amplification in about 40 rounds, you really get crazy amplification. So you need to be able to count original molecules and not the distorted signal after amplification. So every single molecule has its own unique tag. Of course, these are very complex molecular protocols. So again, you can't detect the data face value. And we've developed a pipeline to analyze this data. Another important thing, you're not getting the entire transcriptome, you're sampling it. RNA-seq is sampling, and in this case, you're getting about 3 to 5% of the transcripts in each cell. What do we do with all of this? Now we have single-cell data, high-dimensional single-cell data, single-cell RNA-seq. Well, first of all, again, we want to understand the phenotypic space of cells. We want to be able to understand cell types and cell spaces. So we could look at it from a geometric point of view. You have protein A, protein B, gene A, gene B, and you have 2 cell types here. But as you increase dimensionality, suddenly you see that actually protein or gene Z gives you a distinction, and now I have 4 cell types instead of 2. And these emerging high-dimensional technologies, including single-cell RNA-seq, will allow us to characterize our cell types. The new type of data will be now, again, bringing us into the high big data domain, millions of single cells. There's about 500,000 single cells now measured. Most of this is pretty crappy, but I would say about 100 to 200,000 single cells. But we can now actually get to millions of single cells, and that gives new computational challenges, such as how do we visualize it. And this geometry that I just showed you before is non-linear. So this is PCA, a linear reduction, and you see these are different cell types. Every dot here is an individual cell, and PCA doesn't cut it. It doesn't separate these cell types. Actually, what does separate them very effectively is the TCGA algorithm by Van Mateen and Winton, which we've adapted into VSNE to adapt it to single cells. And you can see here automatically, without knowing anything, this nonlinear dimensionality reduction separates all known cell types without knowing anything. So you see all the different immune subtypes are separated out into distinct regions of the map, visualizing high-dimensional data in 2. Here is an example when you bring it down to 3 dimensions, and you could see, again, cell types are not these nice, round, linear Gaussian balls. They're very, very concave, nonlinear, twisted, geometric objects. And this is 3D, so this is 13 dimensions down to 3. TCGA is very powerful. So here we have a case of healthy blood with a tiny bit of residual ALL. And we see this tiny dot right here. This is a tiny fraction of cancerous ALL cells that are separated out from healthy bone marrow. This works for single-cell RNA-seq. So again, this was a publication, one of the two drop publications I showed earlier. This is a retina with many known cell types. And this is, again, the tsunami map of each individual cell. Again, this is a large feature space, so each cell has thousands of genes. And you only need a small number of reads sampled for each cell. So while bulk RNA-seq, we typically take 30 million reads, here with 50,000 reads, a small fraction sampling, we have enough to distinguish these cell types. But much resolution is lost. We're taking thousands of dimensions and squishing them down into two. That doesn't cut it for clustering, so we developed graph-based clustering algorithms to actually identify and cluster and characterize cell types based on some things that we hijacked from social networks. Each node is a cell. Tell me who your friends are, and I'll tell you who you are. We find communities or social networks of cells. Again, the trick is to build the right graph. Here, the right graph is actually using the Jacquard metric. So you clean up the graph and sort of locally tune K and find and do automatic feature selection by first building a K and N graph based on your favorite distance metric and then automatic selection of keys. This works great. We've been using it in immunotherapy to characterize novel immune subtypes. Our TILs, the immune cells inside the cancer, behave very differently than healthy immune cells flowing inside healthy blood. And now looking at single-cell, high-dimensional exploration of these TILs, we're finding a lot of things that are very surprising in terms of co-receptor expression that actually can help use to guide personalized immunotherapy. And finally, if we have big data, if we collect so many single cells, if we get M, the number of samples, not to be 1,000, but to be 10,000 and 100,000 and 1,000,000, we can actually use the variation in the expression patterns of these single cells to learn networks, to directly learn the circuitry I talked about in the first slide. We do this very effectively in a paper here. I won't go into the details beyond saying that we can learn networks, circuitry, and how it goes wrong in disease just by looking at the variation inside the expression of these individual cells. Again, the trick is M goes up to 10,000 to 100,000. Back to cancer, tumor heterogeneity. We can't look at bulk. Tumors are very heterogeneous. We have many subpopulations of cells, the tumor stem cells, the drug-resistant cells. These are inside an environment interacting with the stroma and these tumor immune cells. And if even 1%, a tenth, 100th of a percent of the cells are going to evade therapy, that tumor is going to come back. And people die not because the first-line therapy that cleared most of the tumor fails, but because the tumor recurs. And we need to really study tumor heterogeneity to look at this 1%, characterize it, identify it, and figure out its vulnerability. And here, single-cell technologies are going to help us with it, characterize, as I said, the drug-resistant populations and the microenvironment. And I want to stress, heterogeneity is not only in the DNA. For instance, Joan Brugge recently showed that you have identical DNA, but the cells that are in interaction with the stroma, that are at a different location, that are getting signals from the stroma, survive P13 therapy because of their interactions with their stroma neighbors. So the next talk we're going to hear about realizing personalized medicine, taking all this big data and saying what drug matches a particular patient. And we definitely need to do that. We definitely need to help our suffering cancer patients and give them the best medicine possible. And with big data techniques, we can and need to do that. And the next talk will talk about that. But this isn't going to be enough. The cancer is smart. Cancer is evolving. And recently it was shown that in most cases, you even have the drug-resistant clone in there when the therapy is applied. So we really need to go towards immunotherapy. Our own immune system is a powerful, evolving, adapting system, and it is capable of attacking the cancer if we coax it. Right now, it only helps a small percent of the patients, but when it helps, it solves it. And I believe the next big push should be big data to personalize immunotherapy. Thank you.